Hello, Prabhakar sir. Hello. Hello. Prabhakar sir. Hello. 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 Prabhakar sir. Hello. 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 Please unmute yourself. Sir, has our resource person joined or not? Just unmute yourself, sir. Yes, yes. Yes, yes, he has joined, sir. Sir, namaskar, sir. Sir, namaskar. Subhendu, sir, namaskar. Hello.
Good afternoon, sir.
Good afternoon.
Good afternoon to all.
No.
Prabhakar sir, shall I start? Now wait one.
Yes, definitely.
So, In the outset, I would like to request all participants, please mute yourself and don't press the sharing button so that we can conduct the session without any disturbance. With the blessings of Lord Jagannath, let me start today's session. Esteemed Principal of Udala College, Udala, revered Chief Guest cum Chief Speaker of today's webinar, Dr. Subhendu Palei sir, respected staff members and students of this college. At the very outset, I, Dr. Prafulla Kumar Panda, IQAC coordinator, express my sincere gratitude to all the participants who have joined from different parts of the country and from abroad as well for your kind support and for your cooperation in these days. We have already organized 10 webinars and this is our first international webinar. So I always seek your participation as well as your cooperation and support. Here I also welcome our Honorable Principal Sir, Professor Gopal Chandra Behra, whose continuous support and cooperation has motivated us to organize a series of webinars. And in this regard, I would like to express my sincere thanks to our revered principal for his guidance and support. I also welcome our respected staff members, students to this session. I also welcome HOD Zoology and convener of today's session, Dr. Prabhakar Sahu sir, and co-convener Ms. Susri Sangita Pati madam. I also welcome assistant coordinator IQAC Mr. Satyavrata Malik sir. I welcome you all to today's webinar on the topic light activated epigenetic regulation of human genome. Further, it's a matter of honor and privilege for me to welcome our honorable speaker, chief speaker of today's session, Dr. Subhendu Palei, postdoctoral researcher, Department of Chemistry and Chemical Biology, Ottohan, Germany. Sir, I on behalf of Principal Udala College and Department of Zoology and the IQAC extend my sincere gratitude and respect and welcome you to this August gathering. Hope sir's deliberation on the chosen topic will be a motivating one, very enlightening one also, and immensely benefit all the participants who have joined from different parts of the country and abroad. And once again, I welcome you all to today's webinar, first international webinar on the very topic that is light activated epigenetic regulation of human genome. Now, as per our agenda, I would like to request our Honorable Principal, Professor Gopal Chandra Behra sir, to give his presidential address. Sir, please. Principal sir, please.
Thank you, Dr. Panda sir. Esteemed resource person, Dr. Subhendu Kumar Palei, Faculty of Chemistry and Chemical Biology, CU, Dortmund University, 44227, Dortmund, Germany. Respected participants, researchers, students of the department, and my respected colleagues. At the outset, I thank you all for joining in our second webinar organized by Department of Zoology in collaboration with IQAC Udala College Udala. In this respect, I extend my heartfelt thanks to the HOD and convener Department of Zoology Dr. P.K. Sahu and a co-convener Ms. Susri Sangita Patti for sincerely organizing this type of international webinar. I also extend my special thanks to IQAC coordinator Dr. Prafula Kumar Panda and assistant IQAC coordinator Satyavrata Malik sir for taking all technical initiatives to upgrade such an international webinar in our college. With an aim to inspire and update our students, teachers, as well as researchers' knowledge during this pandemic situation. It's my pleasure and privilege to express a few words about my college. Udara College Udara was established in the year 1965. The institution became a full-fledged degree college in the year 1971. The Utkal University granted affiliation for honors course of zoology in the year 1988-89. Now this college is running with taking affiliation from Sri Ramchandra Bhanjdev University. Being one of the premier colleges in the district of Mayurbhanj and in Odisha, India, as well as we have total 40 number of honor subjects. Our students have made us proud by securing top position in the university level every year. This college has accredited with B level plus by NAP in the year 2016. Innovation and rejuvenation has become the catchword to excel the future of the college. Our unity and dedication will bridge the gap between mission and achievements. Today's topic is light activated epigenetic regulation of human genome. It's a very current research topic. Our research person, Dr. Subhendu Kumar Pollai. I am very much grateful to Dr. Pollai for his consent as a speaker of this very topic. Now I inaugurate this topic for its discussion and future perspectives. Now more than 1,000 participants, researchers and students in India and outside of India are awaiting for active participation. Thank you one and all.
Thank you, sir. Thank you so much for your support and cooperation, and hope such type of cooperation will continue in the coming days. Now, I would like to request Dr. Prabhakar Sahu, HOD Zoology, convener of this session, to give a brief introduction of the topic. Please, sir. Prabhakar sir, please.
Thank you, Dr. Panda. Thank you. A very good afternoon to one and all. Esteemed Principal, Professor Gopal Chandra Behera, respected Coordinator of IQAC, Dr. P.K. Panda, respected Chief Speaker, Dr. Subhendu Kumar Pollai, respected participants, researchers, respected co-convener of the webinar, Susri Sangita Patti, Assistant Coordinator of IQAC, Professor Satyavrat Malik, teaching and non-teaching staff of my college, and my dear students. Now I am going to talk about the keynote address of the topic, which is entitled Light Activated Epigenetic Regulation of Human Genome. The epigenome defines the unique gene expression patterns and resulting cellular behaviors in different cell types. Epigenome dysregulation has been directly linked to various human diseases. Epigenetic editing enabling genome locus-specific targeting of epigenomic modifiers to directly alter specific local epigenome modifications offers a revolutionary tool for mechanistic studies in epigenome regulation as well as the development of novel epigenome therapies. Inducible and reversible epigenome editing provides unique temporal control critical for understanding the dynamics and kinetics of the epigenome regulation. This summarizes the process in the development of spatio-temporal specific tools using small molecules or light as inducer to achieve the conditional control of epigenome editing and their application in epigenetic research. Chemical modification patterns on histone tails and DNA collectively constitute the epigenome that dictates unique gene expression patterns and resulting phenotypes in each distinct cell type. The alterations of epigenome due to the dysregulation of epigenomic pathways and mutations of the epigenome regulators contributes to pathogenesis of various human diseases, including many developmental diseases and cancers also. Several epigenetic regulatory mechanisms, including DNA methylations, histone tail post-translational modifications, ATP-dependent chromatin modeling, and non-coding DNAs have been shown to play key roles in governing gene activities, genome-wide mapping of epigenome modifications by the next generation sequencing technologies has provided valuable information correlating specific epigenome modifications or combinations with certain transcriptional activities. Today's topic is light activated epigenetic regulation of a human genome. Our research person will broadly describe about the topic, but the theme of this topic again, the 10-11 translocation or TET deoxygenases catalyze the oxidation of 5-methyl cytosine 5-MC to 5-hydroxymethyl 5-HMC, 5-hermyl and 5-carboxycytosine. These nucleobases are not only intermediates of 5-MC demethylation, but can also act as independent regulators of chromatin function. TET mutations are a hallmark of cancers, but how they modulate the dynamics of 5-MC oxidation landscapes is poorly understood and is hampered by the inability to selectively activate individual TETs with temporal control in cells. The research person reports activation of TETs in mammalian cells by incorporation of genetically encoded 4,5-dimethoxy 2-nitrobenzyl L-serine as a transient active site block, and its subsequent deprotection with light. This enables sharing intracellular information of 5-HMC in wild type and cancer-associated TET2 mutants, providing direct insights into the impact of cancer-associated TET2 mutations on the kinetics of TET2 catalysis in vivo and allows time-resolved monitoring of target gene activation and transcriptome reorganization. This sets a basis for dissecting the order and kinetics of chromatin-associated events triggered by TET catalysis, ranging from DNA demethylation to chromatin and transcription regulation. So this is about the keynote address of a large, vast topic. Now, can I hand over the mic to our IPS coordinator, Dr. P. Panda.
Thank you, sir. Thank you for your brief introduction and keynote address. Now, I will request you once again, sir, please introduce our chief guest come chief speaker of today's session, Dr. Subhendu Kumar Pallai.
Okay, thank you. Thank you again. Okay, thank you. It's my honor and privilege to welcome and introduce our keynote speaker of this session, Dr. Subhendu Pallai. At present, Dr. Pallai is continuing his research as postdoctoral researcher Department of Chemistry and Chemical Biology to Dortmund, Ottohan, Germany. His field research is epigenetics, chemical biology. He started his early career as a MS student in chemistry from IIT Kanpur, India. His knowledge and interest for research flew him to Germany, where he completed his PhD in chemical biology at University of Münster. He is the recipient of a prestigious International Graduate School of Chemistry, Münster Fellowship 2012 to pursue PhD. During his PhD, he conducted extensive research on cyclic peptides, bacterial display, and protein engineering. He has expertised on various research techniques like mammalian cell culture, immunofluorescence microscopy, biosulfite sequencing, NGS sequencing, flow cytometry, RT-qPCR, RNA sequencing, cloning, protein expression and purification, SDS phase, Western blot, etc. His research outcomes are highly appreciated and published in many international journals having high impact factors. One of his articles, Bacterial Cell Surface Display of a Semi-synthetic Cyclic Peptide, published in peer-reviewed journal ChemBiochem, it was featured as cover page of the journal and was the most assessed journal article in January-February 2019. He has attended many international conferences where his research outcomes are highly appreciated. He has been awarded EMBL Advanced Training Center Corporate Partnership Program Fellowship to attend EMBO Chemical Biology in 2016 conference in Heidelberg, Germany. He was awarded for Best Poster Presenter in Münster Conference of Biomolecule Analysis in the year 2014 from Springer. Besides these achievements, he has also guided projects of several bachelor's, master degrees, and PhD students for their thesis in chemical biology during doctoral and postdoctoral period. He is not only limited to one research, but is a member of peer reviewer of reputed journals like Pharmaceuticals, Biomedicine, Protein, and Peptide Letters, also provide suggestions to authors on how to improve quality of their manuscripts. He has international affiliations with American Peptide Society from 2019 to till date, European Peptide Society 2019 till date, the Epigenetics Society 2019 till date, and the International Chemical Biology Society 2012 till date. So thank you very much, Dr. Polai, for your achievements and for your consent for choosing this type of college to deliver your presentation. Thank you all.
Now I would request our chief speaker, Dr. Subhendu Kolai sir, please start your presentation. It's your time now. Please, sir, continue. Hello. Hello, Dr. Kolai sir. Hello. 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 Dr. Polai sir. Dr. Subhendu Polai sir. Hello. Am I audible? Hello. Prabhakar sir, please just contact him if he is listening.
Okay, okay,
Try,
okay.
please. Join করଥିଲେ তো? হুম। ଜଏନ୍ କରିଥିଲେ କଟିଗଲା କି କଣ?
Hello, Dr. Ponda.
Sir, sir, has he joined? He has joined.
He has joined but not audible.
Okay. Just give him a message,
So,
sir. Give him a
I have
message.
given the message to him. I have given the message to him. Should I tell him to rejoin again?
Yes, yes. Please, participants, please bear with us. Very soon we will contact and he will deliver his address. He is present. He has joined, sir. Just ask him to continue. He has joined. Dr. Palay, sir. Dr. Palay, sir. Hello. Am I audible, sir? Hello.
Hello.
Dr. Palay, sir. Hello. Dr. Palay, sir.
Hello.
You are audible, sir. But resource person is not audible.
I said to him to rejoin again.
He has joined, sir. It's there. Still, I have just asked him once again. Skip and again he should join.
Hello.
Sir, continue, sir.
Now I can join.
Okay, sir.
Okay. You join and continue. Okay.
Dr. Palay, sir. Dr. Palai, sir? Am I audible, sir? Hello? He's trying, but he's trying to present something, but
He's trying. He's listening to our voice, but his voice is not audible to us.
it's a network issue, I think.
Yes, yes. So participants are requested to bear with us.
Please, sir, once again you ask, he just skip and again he should join. So that would be better.
Okay, okay. Okay.
Okay, sir. Okay, continue, sir. Hello, Subhendu, sir. Am I audible, sir? Yes, yes, you are visible, sir. Now continue. Hello, Subhendu, sir.
Subhendu sir will rejoin and present his topic, okay?
Okay, okay. Hello, Subhendu, sir. Hello. Hello. Hello, Subhendu, sir. Hello. skip reading. ಇದು ಅಚ್ಚುಂಟಿ. Hello, Suvendu sir.
Now Dr. Polay has joined, so he will take time 1 minute only.
Okay, okay. ಜನ ಖುಷಿ ಇಲ್ಲ. He is visible, but his voice is not coming. It's visible. He has joined.
Okay.
Sir, can you hear me?
Okay, okay, sir. Now
Sure. Okay, it's audible. It's audible.
Yes, so I'm joining both from my mobile phone and from the internet, but now I could not join because I have reached 100 participants.
Now you try, sir. Now you try. It's open.
Okay. Yeah, okay. So let me try again. Okay, can you see my screen?
Yes, it's starting, sir. Yes, visible. Now continue, sir.
Okay, can you see my slides?
Yes, sir.
Okay, great. Okay, so I'm audible and you can see my slide, right?
Yes. Okay.
Yeah, so you probably, I don't know if you can see my picture because I'm not sure because I am connecting both from my mobile and from laptop. Okay.
Okay, it's okay.
Okay, so thank you very much, Dr. Sahu sir, for the invitation and kind words of appreciation. Dr. Panda sir and Dr. Behera sir, the Honorable Principal of Udala College, and all the organizers and all the audiences. I'm really overwhelmed to see many people also from different backgrounds in this webinar. 1 minute. Yeah, so am I audible?
Yes.
Yeah. So welcome to today's topic. It's about light activation of epigenetic regulation of human genome. This was the work that I did here in Dortmund as a postdoctoral fellow. And before I start, I would also like to introduce where I live. Can you see my cursor in the screen, Prabhakar sir?
Yes.
Yeah, okay. So I come from the western part of Germany, a city called Dortmund, and you might know who are familiar with football. It has a very famous football club, the Borussia Dortmund, and it's a big city with industrial history. And it has also a very popular Christmas market. It's one of the biggest Christmas tree in Europe. Yeah, and this is my university, the place where I work. And yeah, so this is the Department for Chemistry and Chemical Biology. Okay, I also like to introduce the topic to the speakers because I know to the audience, because I know the audience are from many different backgrounds. So I would like to also be as simple as I can. So before I start, I would like to introduce what is epigenetics. As Dr. Sahu sir already introduced about epigenetic in his beginning words, that epigenetics is the regulation of phenotypes of an organism without changing the genotype. So basically, here is a very simple example. There are two butterflies with different names. Of course, there are different species. One is Papillomachon and one is Iphiclides podalirius, and they have different genomes. So if you can see that they look completely different. This is expected because when you have different genome, you produce different phenotypes. But here is a metamorphosis of the larva to the butterfly, and here you can see that there are also different phenotypes. This is the larval form, and then it metamorphizes to the complete butterfly form. So here, do you think they have totally different genome? It's not. They have the same genome, but they store different phenotype because they have different epigenetic regulation in different stages of their life. So the epigenetic regulation here is completely different from here and then there. So this is a complete new way of controlling the phenotype of an organism. And this is a very simple example, but a very complex organism like eukaryotes, like in mammals, this is much more complex. So all our life, we are born with a single genotype from our parents, but then we store different phenotype in our different cells. For example, the stem cells, you are aware of the stem cells, which are undivided pluripotent cells. In mostly they are found in our bone marrow. You might also know the stem cell transplant of the bone marrow. But what happened when there is the embryo, this stem cell starts to distinguish themselves, and then they reprogram themselves to different body cells. One is muscle cells, blood cells, nerve cell, cardiac cells, liver cells, and intestine cells. And this happens because of the epigenetic regulation, because all these cells of your body have the same genotype. So which means they have the same genome, which all 23 chromosome. If you sequence the genome, there is no difference of the basic letters A, T, C, G, which compose the whole genome of a human. But you might find they have different epigenetic regulation in different cell types. For example, the genes that are activated in nerve cells are not the genes that are activated in cardiac cells, and these are not the genes that are activated in liver cells, because we have almost 25,000 proteins in our body, but we don't need all the proteins to be present in all cells at the same time. So we need different proteins in different cells for different function. And this is how the regulation of epigenetics comes into play. So this is the whole picture of epigenetic regulation, and this is much more complex than the genetic regulations. Yeah. So this is one of the molecular picture that I want to give you. This is much more complex, but I just want to simplify here as epigenetic code. This means the code that is written in your genome and that regulates the epigenetic function. So as you know, the chromosome is a condensed form of DNA. It's supercoiled structure, which is composed of many nucleosomal units, and one nucleosome consists of a histone. You know the histone protein, and this is wrapped by the DNA. And this DNA, if you just look into the DNA double-stranded helix, there are different modifications like DNA methylation. For example, also in histones, there are different modifications, and histone methylation, acetylation, and so on. So the question is what these modifications are doing there. So they are not just the chemical modification, they are to regulate different function. So for example, different histone methylation are activating a gene and different DNA methylation are deactivating. So if there are a lot of DNA methylation in a gene, then the gene is not activated. This means that the gene is not producing the protein that it is supposed to. But if you remove this methyl group, then the gene is active and then it produces the protein. This is what is called epigenetic regulations. So today this is the vast topic, we'll only focus on DNA methylation and the enzyme that is responsible for the DNA methylation. This is another simple picture of how we can read, write and erase the modifications of DNA. So this is a simple double-stranded helix which is unmethylated, and there is an enzyme called DNMT. It's called DNA methyltransferase, which transfer methyl groups from different cofactor and modify the cytosine of DNA. And this is called CPG modification. CPG stands for cytosine and phosphate backbone and guanosine. And this modification is very dynamic, which means that it is once it is placed into the DNA, it can also be removed, or it's called unmethylation or demethylation. And this is called erasing function. So it's like the simple when you write on a piece of paper and you can erase it with a rubber. And this is the enzyme that is kind of erasing the methylation from the DNA, and it's called TET. And TET is standing for 1011 translocase. And there are different TETs, TET1, TET2, TET3. The name nomenclature comes from different, so they are the same family of enzyme, but different way of modulation. I will tell you more about these three enzymes later. So you also know that all our mammalian genome, 70 to 80% of CPG are methylated. So CPG means cytosine, phosphate, and guanine dinucleotide. So wherever you just read the genome and you find CPG, C and G close together, there is a possibility to methylate this position, and almost 70 to 80% are methylated, which means there's a lot of epigenetic regulation going on in our genome with these two enzymes. So DNMT methylate it when there is a not requirement for the activation of gene, and the TET enzymes are then demethylating. This is like turning on and off a gene function in the genome. This is more like a chemical picture. As you know, I come from a chemistry background, so we always would like to look in the molecular understanding of the epigenome. So this is what is called cytosine. You might remember from basic biochemistry classes, and this is the 5 position of cytosine, which is methylated by DNMT, and this is a methyl group. And then there is oxidation by the TET enzyme. Then it forms different intermediates. So, so oxidations mean it will add oxygen, and this is how the next product is formed. It's called hydroxymethyl cytosine, then 5-formyl cytosine, 5-carboxyl cytosine. And then it goes through a repair pathway from this carboxyl cytosine, 5-formyl cytosine, and goes back to the cytosine. So it's complete cycle, and it's complete dynamic cycle. So there is always some balance going on in between, and whenever there is a epigenetic regulation, all these enzymes are playing a very important role. So previously it was thought that it is only the methylation that has a real biological role to play, but currently this field is not so old. So in last decade it has been found that hydroxymethyl cytosine, formyl cytosine, they also have important biological roles to play. So there is a lot to discover in this area. Okay, because I was telling you that the methylation is taking place in the dinucleotide, now you can think of a double-stranded helix. So if there is a CPG here and there is a CPG in the opposite direction, of course, and then there is a methylation that can take place here in the cytosine and here in the cytosine. This means that you have several possibilities of this modifications. So this C can take all of these cytosine, 5-methyl cytosine, hydroxymethyl cytosine, and this C can take any of these modifications. Now you can think so many different types of, almost 15 types of modification that is possible in this single CPG position. So all these modifications might have very important biological role to play, but there is a lot of things that is unknown. So how they are playing a role or not. So basically, to understand these modifications and their dynamics, it's important to know which enzyme is modifying them and how we can control its activity. That brings us to study the TET enzymes. Okay, so this brings me to now the different TETs. So there are three kinds of TET: TET1, TET2, and TET3. So TET1 and TET3 has this CHXC domain. This is the domain that helps the TET enzyme to bind to the methylation chromosome or methylation DNA. But TET2 has no ICXC domain. As you can see, it is not linked. So there is a different partner which is called IDAX, and this is then helping TET2 to bind to chromosome. So there are other domains. So mostly the enzymes are very big, but there is a small catalytic domain which is independently active. So basically, you can just crop this part and use it for your assays because this is itself active. So the other part has a function to play, like binding and other interactions, but this is many of the things are not known yet. Okay. Then we should look more into the crystal structure of this enzyme. So this is the crystal structure of the TET enzyme that is bound to a DNA. As you can see, this is a DNA double helix. And when it binds to the DNA, of course, the aim of this enzyme is to modify a cytosine. Yeah, so here is a cytosine. But how does it modify it? Because the cytosine is in a double-stranded helix. So it is in a Watson-Crick base pair, right? So when it wants to modify a cytosine, it flips out the base. So basically, if you can see, the major groove is now empty. So the base is rotating 180 degree and flips out and then goes into the active site of the enzyme. Now, so the enzyme can modify or oxidize the 5-methyl cytosine. So basically, there are several residues which play important role. One of the residue which is important is the Y, tyrosine. And why tyrosine is important, as you know, the tyrosine is aromatic residue, so it can interact in a pi-pi stacking. So and then it can stabilize the base, and then it can modify the cytosine. Okay. Looking more into details, what are the other components of this active sites? So this is the whole active site of the enzyme. Here is the 5-methyl cytosine. This is a methyl group, as you can see. So basically, there is no residue or active site residue that is doing the oxidation. So who is doing the oxidation? It's the iron. As you can see on this brown spot, this is the iron 2, and it is coordinated to different amino acids. And there is the oxygen also bound to iron 2, and this is the cofactor, N-oxo glutarate. And so basically the iron 2 is then here this is more chemical representation of the oxidation process. This is the active site with iron 2 with bound to histidine, aspartate, and water. So an oxygen comes and then replace this water, and then there is a iron 4 oxidation state, and this then can oxidize 5-methyl cytosine to 5-hydroxymethyl cytosine. So there is not a single residue that is oxidizing, it's the metal, that is iron, that is oxidizing the 5-methyl cytosine. So this is the basic important message for you, that the iron is the main role player of this enzyme. So So what is the importance of this enzyme? Why we are studying it? It has been found recently that there are a lot of TET mutation in hematological cancer, like the blood cancer. So people have sequenced the genome of blood cancer patient, and they found that whenever there is a blood cancer phenotype and or hematological disorder, there is a mutation in TET2, and they then assume that this TET2 mutation somehow is associated with a lot of hematological disease. Yeah, so here you can see that all the patients that have this T-cell lymphoma, chronic myelolytic leukemia, different kinds of lymphoma, of course, they have all these stars, and this means there is a mutation of TEP2. And this is a very important message that when there is a mutation of TEP2, what is happening biochemically? How we can characterize the enzyme biochemically when there is a mutation? Is it really dead, or is it less active, or is it really doing its job or not? So, and another reason to study this TET enzyme is that when there is a embryo that is being developed, a mammalian embryo, and these TET enzymes are playing very, very important role. For example, here you can see the methylation pattern of a cytosine is completely like a reset. Like when you restart your computer, you reset everything. This is what is also being done when there is fertilization. You can see that the paternal and maternal 5-methyl cytosine is decreasing, but the maternal one is decreasing very slowly, and the paternal one is decreasing very fast. So which means the cytosine, the methylation of cytosine is being removed um, by some enzyme, and this enzyme is TET3, which is doing the job in only in paternal chromosome, not in maternal chromosome. This is the most important thing. So maternal chromosome also goes down, but this is like a dilution, which means when the cells divide more and more, then there is a dilution of methylation, and it also goes down. Then this methylation is then coming back during the epiblast stage, and this is done by then DNMT enzyme. The same thing also happening from epiblast to next forms of the embryo, like there is loss of methylation, then there is the enzyme TET1, TET2 coming into play. So as a like a summary of these slides, that TET enzyme are also not only playing a role in disease, but also playing a very important role in development. Because of course, starting from the zygotic stage to blastocyst stage, all of them have the same genome, right? So the gene is same, but how does the embryo knows which genes to turn on or off at different stages of the embryogenesis? This is done by then the epigenetic regulation or methylation. Okay, so now coming to the project that we wanted to design. So basically what we wanted to do is we want to control the activity of the TET enzyme with a cage. So basically, you can think um, this is a like a roadblock for the enzyme. Whenever there is a cage molecule inside the enzyme cannot do its job. So it cannot do the oxidation from 5-methyl cytosine to hydroxymethyl formyl and carboxyl cytosine. This is what I told you before also. So it cannot do its job. But when you remove the cage, then the enzyme is active and it is doing its job. So the cage is now removed, and it can oxidize 5-methyl cytosines to hydroxymethyl formyl and carboxyl cytosines, and then all these bases are involved in regulation of chromatin and transcriptome reorganization. So transcriptome means the gene expression. So how we are doing it, because we are chemical biologists, so we basically use the knowledge of chemistry to introduce the unnatural amino acid. I will talk more about this later. So basically this is amino acid which is not found in nature. So we know that there are 20 natural amino acid in our a natural system, but this is amino acid. This is a derivative of serine, as you can see, but this is modified with a photocase group. So the photocase group can be removed with a blue light. When you just shine a light in a live mammalian cell, then the group is removed, then it forms the serine. And the serine is then the native form where it is found in the enzyme, and then it can do its job. So basically we are blocking the activity of the enzyme by changing a position of serine in the TET enzyme. Now, this is called spatiotemporal control, because with light you can control where and when you want to activate the enzyme. So in principle, this is called spatiotemporal control. And there has been a lot of studies how to control the activity of enzyme with spatiotemporal control with the external stimuli. So basically, with a resolution of space and time. Um, why it is interesting? Because you can use the native biological environment, so the cells are live, it's in situ activation, and there are different kind of stimuli that you can use. You can use light, you can use a chemical ligand like inhibitor, you can use a cytokine, chemokine, or a physical force, for example, like a heat or something, and that it can do its effect on the biological system. But here we are more interested in the light, because light is one of the most effective stimuli, and because it can, you just have to shine the light. There is no need to cut open a cell or anything. It's non-invasive. Yeah, it's very quick because the photons can reach to any point in the cell very quickly. So the delivery is very fast, and you have a very precise control of the area that you want to penetrate, because the light can go in micrometer scale. And there's a low background effect, because most of the biological system are not absorbing the light that you are shining. And it's very fast, so you can sign and remove the source in milliseconds, unlike the small molecule inhibitor. For example, if you want to add it to the cell, then you have to wash it also, and it takes minutes, sometimes to hours. So in comparison to that, this light is very fast, and it can be easily performed with the live cells without interfering with other cellular processes. So it's kind of orthogonal to the living system. That's why light is a very popular tool to control the processes. So now coming to the TET. So we took the mouse TET. So this one is called mouse TET2, and this one has several domains that I also showed you before. It has N-terminal domain and C-terminal domain, which are rich in cysteines. Then the double-stranded beta helix structure. Then the insert, which is low complexity insert, which has no defined structure. And then there is another double-stranded beta helix. And we put different tags. One is flag tag, and another is HA tag. Why we put these tags, you will know later, because when you want to express a protein, you want to also detect it in the cell, and that's why we need these tags. So these are kind of, you can see the ID card for the protein. So you want to detect it, you want to check, okay, this protein has this ID or not. Then we use the unnatural amino acids. This is called 4,5-dimethoxy 2-nitrobenzyl L-serine. So I don't want to go more into the chemistry of this amino acid. You just want to know that when you shine light, this amino acid can cleave here, and it can give to the native serine. So basically, we will get the native serine in a matter of minute, one or 2 minutes, when you just shine the light into the cell. Now the question is, which amino acid of the enzyme we can replace? We cannot replace any, right? So you want to replace one amino acid that is really critical for the activity. So to determine which amino acid is essential for the activity, and if you want to case it, which will block the activity, we tested different strategies to determine this position. So we did modeling studies. So modeling means it's a computational modeling where you replace the residue around the catalytic center with the photocase serine, and then see if this is kind of disturbing or disturbing the structure of the catalytic center. For example, here the S12O3 to 1. So 1 is the unnatural amino acid. Here you can see that it is not disturbing that much. But if you see this threonine amino acid, if you replace with a natural amino acid, photocase serine, you can see there is a large amount of distortion in the catalytic center. And this one is also completely replacing the cofactor if you replace this with a natural. So this is all about in computational studies. Of course, we wanted to test it. If this is true, then we move to the incorporation of this amino acid in cell. Now comes the big important bottleneck of this method. So how to incorporate a unnatural amino acid in a living mammalian system? As you know that our ribosome incorporate the amino acid by protein synthesis, right? So if you remember your lessons in bachelor, that a ribosome is reading a mRNA. So mRNA is transcribed from DNA, and it has different codons. We call it triplet codons, and the ribosome is reading the triplet codons and incorporating different tRNA. And then this tRNA is charged with a natural amino acid. So when there is a triplet codon for leucine, there is a triplet codon for leucine, then it incorporates leucine. When the triplet codon for arginine, it will incorporate arginine. This is how the natural system of translation works. But if you want to incorporate a unnatural amino acid, you have to use its orthogonal tRNA and orthogonal tRNA synthetase. Why? Because if you use unnatural amino acid into a biological system, this amino acid is not recognized by synthetase or tRNA. So basically, it will not be incorporated. But we have evolved a orthogonal synthetase and tRNA, and this is from another organism, and this is from bacteria E. coli. So you just took the E. coli synthetase and tRNA, and then place this to mammalian cell, and this will then recognize the unnatural amino acid photocase serine, and then incorporate this unnatural amino acid in a particular position, and this position is the stop codon UAG. As you know, we have 3 stop codon, and from all of these 3 stop codon, UAG is one of the least used stop codon, and we use this stop codon to incorporate this unnatural amino acid. So basically, you just remember there is a synthetase enzyme which is re-engineered from E. coli to incorporate the photocase serine. So whenever you have this enzyme and tRNA in a mammalian cell, this will incorporate the unnatural amino acid. And of course, you have to add the amino acid to the cells beforehand. So where you want unnatural amino acid, you just replace this codon with a stop codon, and then it will incorporate the amino acid. So this way you can incorporate wherever you want the unnatural amino acid to be incorporated in the protein. And the protein of interest for us is TET protein. And so there has been a lot of studies in this area. So now we can incorporate almost more than 200 unnatural amino acid into mammalian cell, into different organisms. For example, it has been shown in E. coli, bacteria, yeast, C. elegans, zebrafish, mouse. So you can incorporate so many unnatural amino acids in all these organisms, and you can study so many biological functions. So this is called genetic code expansion technology, because we can expand the genetic code from this 61 sense codon, right? Okay, so we wanted to test if this incorporation is working with the system that we have. So we use the mCherry GFP system, where you have a Y39. So a tyrosine is replaced with a TAG stop codon. This means if you use the orthogonal synthetase and tRNA and the amino acid, it should then incorporate it here, and you have expression of GFP. If there is no incorporation of unnatural amino acid, you don't see the GFP express, you only see the mCherry express. And mCherry is a fluorescent protein, and GFP is also a fluorescent protein, which you can see in the microscope then. So here you can see that without the unnatural amino acid, there is no expression of GFP, which means it is not incorporated, and with the unnatural amino acid, you can see GFP is expressed, which means the unnatural amino acid is incorporated into this model protein. And this you can see also with FACS. So FACS is flow cytometry, where you can count single cells in the flow cytometry. Okay. Then we wanted to incorporate this unnatural amino acid in the TET2 protein, and we choose, as I saw you, that these three positions, S1203, T1285, and S1812, and they incorporated unnatural amino acid one at a time in all these three positions. Then we wanted to detect if the unnatural amino acid is incorporated or not. And here there is no frozen protein. What we did was then putting a tag on the C terminal. So if there is incorporation at either of the three positions, then this protein is synthesized and you have expression of this HA tag. And then you can detect this HA tag with a protocol called immunostaining. So just to make it a little bit clear, I have made a little cartoon. So immunostaining is you detect the epitope, like a tag here, with a primary antibody and a secondary antibody. So you know antibodies that are detecting this tag, and then the secondary antibody is labeled with a fluorophore, like a fluorescent molecule, and you can see this whole complex in FACS flow cytometry. Yeah, and you can see that we could successfully incorporate the amino acid in all the three positions. So without unnatural amino acid, you don't see the expression of protein or low expression of the full length protein, and with the unnatural amino acid, there is a higher expression of full length protein. So this is also depicted in this graph, where you could incorporate the unnatural amino acid in different positions. Okay. Yeah, so this is the whole protocol, how we do it. Like you fix the cell, then you incubate with different antibody, and then you could do the immunostaining. So it's a long protocol. So now, after the successful incorporation of unnatural amino acid in different position, we wanted to do a photoactivation with light. So you shine light with the unnatural amino acid being incorporated, and you want to detect if the enzyme is active or not. And to detect if the enzyme is active or not, we have to detect the product of the enzyme. And if you remember, I want to show you what was the product of the enzyme. So here, so the enzyme is doing its oxidation from 5-MC to HMC, and we should detect the HMC in the human genome. And this is what we want to detect after the activity of the enzyme. And to detect it, we use the same immunostaining protocol. So basically, instead of HA tag here, and then you are detecting the 5-hydroxymethyl cytosine with the antibody. So there is an antibody which can detect this modification in human genome, and this is then done by flow cytometry. That with light you can see that there is a formation of product. For example, there is a percentage increase from 5 to 15, 1 to 21, 2.3 to 29.8, which means there is an increase of 5-hydroxymethyl cytosine by shining light. So the enzyme is active basically by shining blue light. This clips off, and you have the serine, and the enzyme is active. This is also what you see here. So in all the three positions, the S1812 position is one of the most efficient, because you can see that there is a almost 15 14 fold increase of the 5 HMC positive cells by light activation. So we use this mutation for further study, because this is one of the best working mutation in our case. And this is another way of looking the photoactivation of TET2. So here what we do is you do the same immunostaining, but then you instead of doing fax or flow cytometry analysis, you just use a microscope and see if which cells are fluorescent or which cells are not. So these are untransfected cells. So basically you don't see no HMC, not TET2 expression, nothing. And there is no effect of light also in the untransfected cells. And these are the HEC cells or human embryonic kidney cells. These are kind of model cancer cells. And then there is a wild type TET, which means this is a TET that has been not caged. It's fully active. So it has no dependence of light. So even without light, you can see that the enzyme is transfected. You can see the expression of the enzyme with this color. And there is a formation of HMC or hydroxymethyl cytosine, which is a product of the oxidation process. But in the inactive enzyme, so in the mutant enzyme, which is fully inactive, so there is no activity of the enzyme, you can see that it's transfected with the transfection control, and you can see the enzyme is expressed, fully expressed, but there is no activity seen, which means the enzyme is inactive. So basically, these are the two extreme scenario. When you have activity, you can see this green color, which is a sign for HMC formation. And when there is completely dark, which means you don't see HMC formation or no formation of product hydroxymethyl cytosine. This is what you see here in this picture, that with all these three mutations, with the light sign, you see that the formation of green color. And this green color is coming from the immunostaining of hydroxymethyl cytosine, which then refers to the activity of the enzyme. So whenever you shine light, the enzyme is active and you can see the green color. And when it is not signed with light, you don't see anything. So now we can successfully cage and then decage the activity of TET enzyme with light. This is the first step of this project. Then what we wanted to do is then to measure the kinetics of HMC formation. So basically, it is currently not possible to characterize the rate constant for this enzyme because we don't have a starting point. Because if you don't use a light, if you just transfect, or when I say transfect, it means scientifically to put a DNA of TET2 into the cell. So when you put a DNA of TET2 into the cell, it will start expressing, and there is no starting point. So it's like you think of a race as a marathon race when there is no starting point, so you don't know who is winning or losing. So this point, when we photocase the enzyme, what we did, we have a defined starting point. So we can set the trigger with a blue light and then it starts to go off. So basically, then we have a point 0 where you can start with a blue light, and then time after light activation, you can measure the HMC formation over time. And you can see that without light, this gray dots, that you don't see activation of enzyme. So there are no HMC positive cells formed even after 24 hours. But with blue light, with this black dots, you can see there is a continuous increase of HMC formation. This means that we can now plot this in a linear form and find that the rate constant of this enzyme is 1.25 hour inverse. So this is like we can precisely characterize in live cell the activity of TET enzyme with this technique. And this was all done in mouse TET2. Of course, we then also control how much exposure time we need to give. You can already start activating the enzyme just with 30 seconds of light, signing light. So if you sign the blue light just for 30 seconds, it is still is active. You can see that the percentage of HMC positive cells from this. And also you can titrate different amount of unnatural amino acid, how much you can give, because too much of unnatural amino acid will be toxic to the cell. Okay, so then we shifted our focus to human TET2 because human TET2 TET is one of the most relevant protein that we wanted to study because they have very important roles to play in disease and development, and it's of course related to human health. As you can see, if you just do a alignment of mouse and human TET, there are a lot of similarities, except this region which is called low complexity region, and this is not necessary for the activity. So the residue that is we are targeting for the photoactivation is here, the serine residue, and this is all same for all three TETs. So basically we can use the same position for the incorporation of unnatural amino acid. So we moved forward with this and we incorporated this unnatural amino acid in all three human TET, human TET1, human TET2, and human TET3. And you can see that if you incorporate an unnatural amino acid and then you shine light, you can see there is a light dependent activation of TET activity. So with this position in TET1 and this position in TET2 and this position in TET3, you can see there is a light dependent TET activity. Whenever you shine light, there is a increase of HMC level. And this is also evident from this immunostaining, where we again did this immunostaining with, of course, wild type inactive TET. You can see in wild type there is a activity of TET enzyme, but when you case with photocase amino acid without light, there is no formation of HMC, hydroxymethylcytosine, and with light there is a formation of HMC, and this you can see in all three TETs. Now, what is the use of this photocasing technique in human TET? We then use it to precisely determine the kinetics also of human TET. So here is the comparison, what I was telling you. If you don't case a human TET and if you case a human TET, what is the behavior of the HMC positive cells? So the behavior of the oxidation. Here you can see once you do the transfection or you just put the DNA into cell, for almost 8 hours, there is no formation of the enzyme, because once you transfect a DNA into a cell, it will start producing the mRNA, and the mRNA will be recruited, and all this translation machinery will come, and then it will express the protein. The protein will fold. This is the whole procedure that needs to takes place before the protein is active. So the protein will fold and the protein will do a post-translational modification, and all this process will then affect the activity of the protein. That's why you see a dim, like no activity in the beginning, and there is a sudden increase of activity after 8 to 10 hours. But if you use photocase strategy, so basically all these processes are done, and then the enzyme is just waiting for the trigger from you to shine a light. So the enzyme is just expressed doing the folding. Post-translational modification is also done. It is almost 48 hours after this incorporation, and then you just shine the light, and then the actual enzyme starts doing its job. So then it's active, and then it's you see that a formation of the oxidized product 5-hydroxymethylcytosine. So here the difference is you are only now measuring the activity of the enzyme. There is no interference from expression, no interference from folding, no interference from post-translational modification. So all these processes are not interfering with your measurement of kinetic of the enzyme. So you see a strict linear kinetics with a rate constant of 1.23 hour inverse. So this is the main difference of the photocase strategy and non-case strategy. We can use it for precisely determining the kinetic rate of the enzyme. So you would also see that the expression or the HA positive cells, which means the amount of the photocase enzyme is always remaining constant during this photocasing process. So you can see the starting from this 0 hour to 24 hour after shining light, the enzyme is remaining constant, and this is because you have removed the unnatural amino acid from the medium, and the cells is having no unnatural amino acid. So the expression process is stopped, because when you have no unnatural amino acid, the enzyme is not expressing at all. Okay, now to apply this precise in vivo kinetics determination method, we wanted to test several cancer-associated PET mutants. So these are the mutants that are mostly observed in as a nonsense mutation in hematopoietic cancer cells. And from all these mutations, 35% of mutations are actually missense mutation, and which means missense means it will incorporate a different amino acid. So it will not completely kills off the enzyme, it will incorporate different amino acid, and it is not completely known what's the amino acid doing, if it is slowing down the enzyme, if it is completely deactivating the enzyme or not. So we choose from this several mutation, we choose few of them, and these mutations are depicted around the activation site, because we thought that these activation which are near to the activation site can modulate the activity directly, because if there is a mutation here, it will affect the binding of cofactor and so on and so forth. Then we move. So these are the mutation that we studied, and this is from a cosmic database. So there's a database for all these mutations where you can also go and look like which mutation was found when and which disease. For example, S1286 was found in a skin, head, and neck cancer tissue, and for example, this P1617H was found in hematopoietic cancer. And there are different publications and how many times they have been reported. So when it says 1, 6, or 2, which means they have been reported in these many patients. Then we could do our photocase strategy with all these mutation. So wild type means it's a wild type TET with no cancer mutation, and all these are the cancer mutations that were incorporated, and with photocase strategy we could precisely determine their kinetic and see that some mutations are completely deactivating. For example, this Y1902S, H, with light activation, you don't see any increase of HMs. But there are some mutations which are not affecting at all. For example, these two mutations, and there are some mutations which are kind of intermediary affecting. So they're affecting the activity, but not so high. So there is a medium activity still remaining. So this is now completely clear. The cancer mutations are behaving in different ways in different mutant. And the reason for the inactivity you can see now in the crystal structure. For example, if you remember, this Y1902S and 8 mutations are completely deactivating. The reason is, if you see this tyrosine residue here, it is very close to the 5-methyl cytosine residue. So it is involved in stabilizing the base and doing this oxidation process. But if you just mutate this now to any other residue, this stabilization is gone, and then the enzyme is completely inactive. That's why these mutations are very fatal to the enzyme. Similarly, there are other mutations which are also affecting in similar way. Okay, then we wanted to measure precisely the rate construct of the mutants. So we choose one of the 4 mutations, S1286, 1286P. You can see that the K observed of these two, so the rate constant of these two mutations are much less than the wild type enzyme, which is 0.73 and 0.43 hour inverse. So these mutations basically slow down the enzyme and the oxidation process. And there are also other mutations which are not affecting at all. For example, this A1505, 16, 17, you can see they are as good as the wild type enzyme. So they are slowing down in the beginning, but they are at the end of 24 hours, they are as good as the wild type. So there is a different kinetic path that is being taken from these two mutations. So it's very interesting to see how different mutations behave differently in cell. So this is the first time we could determine the live cell in vivo kinetics of a cancer-associated mutant in HEX cells. Then we move to determine how this if the light activation tool can be used to control a gene activation. As you know, as I introduced you previously, that the methylation and demethylation pattern can control the activity of a gene, because when you remove a methyl group, then this is activating a gene, and when you put a methyl group, this is kind of suppressing a gene expression. And this is what you also see here. For example, this is a ARC gene. As a, it's called activity regulatory cytoskeleton. So all this cytoskeleton in our cell is controlled by this gene. Yeah, so it's very important for a cell to have the cytoskeleton. Without this, cell cannot survive. So here you can see with inactive TET, if the TET is not active, the expression of this gene is reduced. But when a TET is active, the expression is increased. And this is the same thing that you also see without light, the expression is reduced, and with light, the expression is increased. So we can control the expression of ARC gene with light. And then we wanted to also measure the kinetic of this expression. As you see that over time, after shining light, there is a continuous increase of gene expression of this gene. So you can now also see that you previously saw the activity of the enzyme by measuring the 5-hydroxymethyl cytosine increase, but this will also result in the expression of different genes, because these bases are not just they are just for modification, but also controlling different epigenetic process. And this is one of the example how you can control the expression of a gene after activating a TET with light. Then we went to further deeper into this by controlling the light activation, how we can control different gene expression, not just one gene. We wanted to see what are the other genes in the whole cell. So as I said, there are 25,000 proteins in our human cell. Of course, to know it, you cannot just use a RT-qPCR, which is a PCR technique, but you have to use a different technique, and this is called RNA-seq. So it's a RNA sequencing. So what I did, we did the light activation from here, for example, and then we control the sample after 4 hours, after 8 hours, and then send this sample for sequencing. So we extract the RNA from this sample, and the RNA would be sent for sequencing. So what is the RNA referring to? So a RNA is referring to the expression level of a gene. So if you have more RNA, then you are having more expression of the gene, because DNA doesn't express a protein, right? And it first expressed to RNA, and the RNA express the protein. So what we are doing here, we are measuring the level of RNA by RNA sequencing, and thereby determining the gene expression by our method. So here you can see that there is a difference between different time points. So it is called differential gene expression, where in 4 hour versus 0 hour. So 0 hour means there has been no sign of light. So there should not be less or very few gene expression. And with 4 hour, there is a huge sign of light or after 4 hours. So you should expect more gene to be activated. So here you can see that so minus means down regulation of genes, and plus means up regulation of genes. So with 4 hour versus 0 hour, there is a difference of down and up regulation. So some genes are activated and some genes are deactivated. Similarly, with 0 hours and 8 hours, there is also a huge difference, because it takes more time. So more HNC are formed and more genes are activated or deactivated. And there is also a few difference in 4 hours and 8 hours. To know which genes are there or not, we then look into individual genes. So before I move to the genes that are activated or not, I also wanted to discuss that there are genes which are called stably expressed genes. So these are the genes which people know that there should not be changing the expression with respect to the epigenetic change. So they are very stable, and this is like a control. You can see that with your RNA sequencing, these genes are not changing. So there is no differential regulation of these genes. That's why it says not significant. So there is no difference either you sign right or not. These genes are not changed. So this is also one of the positive control that we have. So now it comes that you can see these are the genes that are being activated. So the more red thing means it is more activating. So the expression is going up. And this is a 0 hour, which means there were very low expression. After 4 hours, some genes are mildly expressed and some genes are very strongly expressed. And after 8 hours, there are more strong expression. But there is also some genes which are also expressing after 4 hours, then being suppressed after 8 hours. So this is complete complex dynamics of this gene expression. One of the most important thing is, like you would ask, how do you know that the TET is the reason for the expression of the genes? We know it because 65% of the genes were found to be differentially regulated, also have a TET signal. So this green thing here means when there is a differential regulation, if this gene is being expressed, we also found that the TET is occupied in the promoter region of this gene, which means it is activated or deactivated depending on the occupancy of TET. So the TET is a reason for the differential regulation of genes. So almost 65% of the genes were found to be occupied by TET enzymes. So the TET enzyme binds to this promoter of this gene. And the genes are categorized in different like different section of the biochemical process. You can say this is mostly related to transcription regulation, RNA processing, N6 adenosine methylations, and chromosome regulation. So all these processes are involved in epigenetic regulation. So this means after light activation, you are affecting those genes that are involved in epigenetic regulation, and these epigenetic regulations are these ones: transcription, RNA processing, or chromosomal regulation. Of course, you can go deeper to look individual genes which are affected. For example, here we also upregulate, so there is an increase of expression of this gene H3C14, which is involved in histone H3 methylation. And we also activate genes EPC2, which play a role in transcription or DNA repair, and so on and so forth. So if you categorize these genes, this is a simple color coding which I gave. There is a genes for transcriptional regulation, genes for RNA processing, genes for chromatin regulation. All the genes are being activated with light activation. But here I have specified there are some genes which are not only activated but also down regulated after 8 hours. So when you shine light, they are activated. Then after some time, after 4 hours, they are also down regulated. This IRS4 RCN gene, they are basically also down regulated. So it's not always true that you are only activating a gene. You can also deactivate a gene depending on the dynamic of this gene. So basically there is very complex dynamics that is being involved in this process, in this epigenetic regulation. And this you could never be able to know unless you use the light activation tool, because here now you have a precise time point of 0, 4, and 8 hours. You can measure at each time point the activation of gene. If you don't use this tool, you cannot see what is going on. You will only see a starting point, and after 1 day or so, an end point. And what went wrong in between, you will never able to know. And so with this tool, we are able to know there is also a different layer of complexity in this gene expression. Okay, now we came to my summary and outlook of this method. So we could activate the TET dioxygenase with light, with a precise spatiotemporal control. And we could first time measure the in vivo kinetics of TET catalysis, so the activity of TET in cell, in live cell. We could characterize the cancer mutant, which gave us a deeper insight to look into the cancer mutations and to understand the enzymes. And we could also do a time resolved monitoring of gene expression, a targeted gene expression, and also a transcriptome reorganization. So we could see that how the transcriptome is reorganized with different dynamic, different dynamic of epigenetic regulation. Now I came to acknowledge my whole group, of course, my professor, Professor Daniel Zummerer, and our collaborators for the contribution to this project. I also like to thank my institute, Chemistry and Chemical Biology, and TU Dortmund University, and most importantly, the funding from ERC in European Research Council, a grant EPICORD, which is involved in this project. And of course, I would like to thank Udala College and Department of Zoology, and Dr. Sahu sir, Dr. Ponda sir, and Dr. Behera sir for the kind invitation. And it's my pleasure to have some questions from you. Thank you very much.
Thank you, sir. Thank you for your nice presentation. Really, it's a very impressive one and a lot of informations that will be useful for our scholars as well as academicians and faculty members. Really, we are quite indebted for such a nice presentation, sir. Now it's a time for question answer session to continue. Now I would like to request Sushri ma'am, please, if you have any question put by our participants, please ask our resource person, please.
Thank you, sir. Thank you, Dr. Malik, for your very informative presentation. There are some interesting queries asked by our participants. On behalf of them, I am quoting a few of the questions. One of the very interesting questions asked by one of our participants: what is the technique to know whether a gene is methylated or not?
Yeah, this is a really interesting question. So I didn't cover this in this part. So there are very several different techniques to know if the gene is methylated. So I want to distinguish this from usual sequencing technique. So if you want to know a gene, what you do is a sequencing. Yeah. So you read different letter codes A, T, C, G, then you know the gene is this one. But if you want to know the modifications, if the gene is methylated or not, the sequencing is not enough, because the modification is not involved in the Watson-Crick base pairing. So basically, you have to use a different technique, and this is called bisulfite sequencing. And this sequencing involves modification of the bases into simpler form, and then detect this with the Watson base pairing. So this I have not covered, but this is another topic to discuss about the sequencing. But as you, this will be a different approach. So it's called bisulfite sequencing for the determination of methylation. I mean, there are also other techniques like nanopore sequencing and TET assisted sequencing to know all these modifications. So there is a, just you know that there is a specific sequencing method to know this methylation, if the gene is methylated or not.
Okay, thank you, sir. Another question: another question asked by one of our students.
Yeah.
Why cytosine is the
Mhm.
preferred site of methylation, not other bases?
There, it's not true. I have talked about cytosine here because it's one of the most widespread methylation, but there are also other bases which are also being methylated, like adenosine, as I showed you this presentation. This sixth position is also being methylated, and fourth position of cytosine is also methylated. And so there are also other bases which can be methylated, but cytosine is one of the most prevalent base that is being methylated. And also, it's not only DNA that is methylated; it's also RNA bases that are methylated. So it is much more complex. But here, I was only talking about DNA methylation.
Okay, sir. Thank you. Should I proceed to next question?
Yeah.
Okay. So next question is, why only blue light is used for photo activation of M state to CD? CT
Yeah.
to CD.
So yeah. So we use blue light. Maybe I should share that presentation. So we use blue light because the photocase group that I am showing you here. So the photocase group is sensitive only to this light, blue light of 365 nanometers. So it is coming in the range of UV region, and of course, it's like a signal and a receptor, right? So if you have a receptor, this is here the photocase group, and you have a signal that is a light, and they can interact because of this. So the blue light is only activating and cleaving this bond. If we use another light, then it's not activating. And there is a very nice thing about it, because we have to work in a normal light condition. As you know, our visible light is ranging from 400 to 700, right? The sunlight. And if you want to use any other light, let's say a green light of 500 nanometer, then it will interfere with this technique. So basically, you have to work all the time in the dark to avoid this accidental photocasing. Because we use blue light, which is not common in the environment, we can only shine this with a particular light source. This is very useful for us it's orthogonal to the biological system. So you can only activate it when you expose it to a particular wavelength. So you can work in the light, handle the cells, do all the experiment with normal sunlight. So there is no problem. So this is the reason that the blue light was used.
Okay, sir. Thank you. And the next question is asked by Yash Kumar Jaiswal. What is the main challenges for light activated epigenetic regulation of human genome in this time? I think I should
What is the
modify.
main challenges?
I think I should modify the question. What are the main challenges you face
Hmm?
in your during your research for light activated epigenetic regulation of human genome?
Okay. Okay. So what I face was, of course, as I showed you here, that first you have to know which amino acid you want to use for the light activation, because this is very critical. And then the incorporation of the amino acid. So the enzyme that you choose, you have to incorporate the natural amino acid into the enzyme. So you have to also find the proper position where you want to incorporate, because the enzyme is 1800 amino acid long. It's a huge protein. You cannot incorporate anywhere. You have to find a particular position, and this then we had to determine the caging position. Of course, after the determination of caging position, another important hurdle was to incorporate the natural amino acid and then see the effect of this incorporation with light. So we could then see, so this was the main bottleneck of the project. And once we could activate this with light, then we could do a lot of biological study. Yeah, so I would say this was the main challenge to introduce a unnatural amino acid into the biological system. Because this is not done with simple protein engineering. It has to be done with also genetic code expansion. It's a new area of biology. It's called more like a synthetic biology, where you incorporate an unnatural system into the biological system.
Okay, sir. The next question is, as you said, TET mutation is related to various cancers. And
Yeah,
TET, we
mostly.
know it is related to mostly blood cancer. So TET enzyme is required for demethylation. And we know
Yeah.
demethylation is required for gene expression. So the question is, are the TET enzymes whether the TET enzymes are related for tumor suppressor gene activation?
TET enzymes are related to tumor suppression gene
Suppressor
activation.
gene activation.
Um, so as I said, the demethylation is of course in many cases activating. But if you remember my last RNA-seq result, there were also some genes, not so many, which are being not only expressed but being suppressed. For example, here you can see there were some genes after 8 hours of light, they were also suppressed. And the same thing also here you can see that most of the genes were upregulated here, the fold change is plus, but there were some genes which were also downregulated. So yes, there can be some genes which are downregulated or suppressed depending on the demethylation. So this is true, yes. But I can't say it's a tumor because this was done in normal hex cells. So of course, in different cell type, this behavior can change. But yes, there can be a possibility of some genes being suppressed. And that can be the reason for a tumor or not. And this is something a matter of further study, I would say.
Okay, sir. Then okay, one question asked by Deepika Rana. Uh, can you please tell us about some other nucleo-based modifiers apart from TET?
Yes, there's so many modifiers as I showed you in the beginning. So nucleobase modifier wise the DNMT, which is kind of introducing the modification, and then TET enzyme, which is removing the modification. In TET there are TET1, TET2, and TET3, and there are also different enzymes for different modification in RNA, which are also removing the modification from RNA base. Yeah, so there can be different modifiers, and because I was only focused on DNA modifiers, I only introduced TET enzyme in this prospect. But there are also other modifiers which recognize different base and oxidize them.
Thank you, sir. There is another question. Can it be possible to reverse the mutation of TET2 in living cells to cure cancer?
Yes, this is not a research area of mine, but this is something that you also hear in the news. This is called genome editing, right? So also Dr. Sahu sir also introduced this topic bit in the beginning, that you can change the genomic composition of this mutation by different technique, and one of the most popular one, which got recently Nobel Prize in 2020, is called CRISPR-Cas9. You might know this, that you can introduce any mutation into a genome, including mammalian genome, and cure different diseases. And one of these also can be cancer. But this has not been practiced successfully in the healthcare. There will be some more research that is needed to introduce to this healthcare system, because we need a lot of other controls like trials and to introduce this. But yes, theoretically it's possible to correct the gene from a cancer cell. Yes.
Okay, I think there is no more questions.
One question is here,
Okay, thank
ma'am.
you.
One question is here, ma'am. Asked by
Yeah.
Santosh Patra, whether any genomic homologs is there between human and pangolin species.
Okay,
Whether
this I am not sure. So what is pangolin species?
I don't know actually. It's
Is it? So if it is a mammal, so of course most of the eukaryotes have this PET oxidizing enzymes. So only it's not present in the prokaryotes like bacteria and all. So there must be a oxidizer PET dioxygenase homolog in eukaryotes. So I'm not sure the pangolin organism, if it is a eukaryote or bird or something, there is definitely a homolog. That's true. Yes. But I don't know this for sure, because I am not into the pangolin thing yet. So, but one can always do some research on it.
okay, sir. Thank you, sir, for your nice answers. Now I would
Yeah,
like to.
you're most welcome, sir.
Now I would like to invite Susri ma'am to present a vote of thanks. Susri ma'am, please.
Okay, sir. Very good afternoon to one and all. Myself Susri Sangita Patti, lecturer in Zoology, Department of Zoology, Udara College, Udara. I deem it a great honor and privilege to propose the vote of thanks on behalf of Udara College and Department of Zoology. I convey sincere gratitude to our Honorable Principal Sir, Professor Gopal Chandra Behra, for giving us permission to organize this webinar, and also for his constant encouragement. Thank you, sir. I extend my heartfelt thanks to our today's speaker, Honorable Dr. Subhendu Palai, for enlightening us with his knowledge and research experience. Sir, indeed this was a very informative discussion. Today's talk not only gave us deep insights to the topic, but also revealed many interesting facts. Once again, I thank you, sir, for sparing time from your busy schedule to grace the event. I also thank our head of the department, Dr. Prabhakar Sahu, sir, whose effort and willingness has made this webinar successful. I mention my deepest sense of appreciation to Dr. Prafulla Kumar Panda, sir, Coordinator IQAC, for all his support in every possible way. I also thank Professor Satyavrata Mallik, Assistant Coordinator IQAC, for his cooperation. The Department of Zoology and the IQAC is thankful to all the participants present here for accepting our invitation and extending their support throughout the session. Last but not the least, I appreciate and thank all teaching and non-teaching staff members of the college for their support and cooperation behind the screen. Once again, I thank each and everyone for their support and cooperation in making today's event a resounding success. Thank you very much for your kind attention. Thank you all.
Thank you, ma'am. Thank you, Subhendu sir. Thank you. Namaskar. Namaskar, sir.
Thank you. You are most welcome, sir. It's my pleasure. Namaskar.
Okay, thank you all the participants. I have already posted the feedback link here as well as there in YouTube. So please fill up it correctly and wait for 2-3 days so that I could generate all the certificates and give you the link in the groups. Thank you once again. Thank you, Subhendu sir. Thank you, HOD Zoology Prabhakar sir and all the staff members here and students and my loving principal, Professor Gopal Chandra Behra sir. Thank you once again. Here I let me wind up the session. Thank you again.
Thank you. Thank you.